starting now. Good evening, everyone, and welcome to the second session of Meet the Guru. And today's guru, as all of you would know, is Dr. Thomas Poole. Dr. Thomas Poole needs no introduction. I'll just give a very small brief about him. He is a world-renowned embryologist and director of the Austin Fertility and Reproductive Medicine. He's also the founding member of the Fertility Center of San Antonio. Dr. Poole is a pioneer in the field of embryology and has been integral to the development of culture media in which embryos grow in IVF laboratory. Dr. Poole is a charter member of the College of Reproductive Biology and American Association of Bioanalysts. He's on the board of directors of the Pacific Coast Reproductive Society and is an active inspector in the Reproductive Laboratory Accreditation Program of the College of American Pathologists. He has been both certified as a high complexity clinical laboratory director through the American Board of Bioanalysts for over 22 years. He was the 2018 recipient of the Lifetime Achievement Award presented by the Society of Reproductive Biologists and Technologists, an affiliated society of ASRM. He has written and co-authored over 100 research publications, books, chapters, review, articles, and abstracts. He's been a frequent speaker in postgraduate courses for the American Society of Reproductive Medicine, the College of Reproductive Biology, the European Society for Human Reproduction and Embryology, along with conferences and symposia throughout North America, South America, Europe, and Asia. And in conversation with our guru, Dr. Poole, we have Dr. Sayali. Dr. Sayali, she has completed her MS and PhD in reproductive biology from Northwestern University, Chicago. She is PG certified in genetics and holds a certification in clinical embryology from Cleveland Clinic. She has done her PGD, PGS embryo biopsy certification course from Embryo to Spain and obtained her PG certification in fertility preservation from the American Society of Reproductive Medicine. She is affiliated as a research scholar with Ronin Institute. She is the Chief Scientific Officer with Cellsure Biotech and Research Center in Mumbai and a consultant embryologist for many IVF centers across India and director at Reproductive LLP. She is also on the advisory panels of Embryonics AI Private Limited in Israel and India and Infertility Division for Shield Healthcare Private Limited. She has over 50 presentations in national and international conferences as a speaker and has also co-authored books like The Clinical Guidelines for IVF Practitioners and Current Indications and Legal Implications for 2020 for IFF. So Dr. Sayali, we are waiting for you to start the conversation with our guru, Dr. Poole. I would request all the participants, whoever, if you have any questions, things that you would like to know from Dr. Poole, which will inspire you and you can learn from them, please feel free to post in the Q&A window. And Dr. Sayali would be taking those questions during the interview. Thank you. Over to you, Dr. Sayali.
Hello. Good evening, everybody, and good morning, good afternoon, wherever you are globally. It's a pleasure and an honor to be able to have a one-on-one with Dr. Rusty Poole. He's one of the idols for us when we had begun embryology training. And I am very excited that I'm being able to ask all these questions to him regarding his career and hoping to get inspiration from him. So, yes. Right. So, Dr. Poole, can you hear me?
I can.
Hi.
Yeah. Hi.
Yeah. So, before I begin, I just wanted to reiterate what a big fan I am of you and all your work in embryology. Being studied in USA, you are one of the names that we always used to refer to and learn from, right from your chapter on troubleshooting to your all the work that you have done in laboratory accreditation. So that was something that was exciting to us. So without further delay, I want to start with the one-on-one and begin by asking questions.
Okay.
The first question is, can you describe how your childhood was in Texas? Because I can see from your CV and from all your talks that you're really connected to San Antonio and to Texas. How is your native place and how has it influenced you in your career, personally and professionally?
Yeah, it's hard to get Texas out of your blood if you're from here. Uh, we say it's hard to get it off your boots. That's the problem. I was actually born and raised in Houston, a large city, but I had my mother was from a very small town in the middle of Texas. My dad from Waco, and in, with numerous relatives in the rural areas of Texas. So I had an exposure to both sides, big city, and spent a lot of time in small communities, farming, ranching communities. I had relatives that were farmers and ranchers, and so we got to spend quite a bit of time in and out of the city. Really very straightforward. I attended a good high school in Houston. Got a good education. I was not a very good student. I wasn't interested in much of anything other than just agriculture at that time. But it was a good solid background. And great parents. I had two sisters, and it was a pretty uneventful childhood, but it was one that we stayed, roamed around Texas on vacations and whatnot, and participated in a lot of kind of standard Texas activities. So it was pretty straightforward. And well, if you can see from this slide, fundamentally, when I was in high school and early college, I was basically just a cowboy. I studied agriculture at a small university in Texas, but weekends there was a collection of us that just hit every rodeo we could get into. This is actually a rodeo in a small town outside of Houston, and that's my best side, the backside there. But I used to.
That looks really dangerous.
Well, I used to ride bucking horses and bull riding as well. And I wasn't very good at bull riding. I won a little bit of money riding bucking horses, but that eventually wore off, and I came to my senses and started kind of buckling down and studying when I was in college. So it's kind of just a very standard background.
Yeah, I think a loss for the rodeos, but a big win for the IVF community.
Yeah.
Yeah. So, yeah, speaking about your basic education, and when you start buckling down, you joined Dr. Terry Hoge from San Jose State.
Yes.
Yeah. How did he impact your research?
Well, as an agriculture major, animal science major initially, I had to minor in biology or chemistry, so I decided to minor in biology. And I was really fortunate. I mean, that's kind of everything that's happened to me, being in the right place at the right time, meeting the right people. It's just been a lot of luck. But I had a really good zoology instructor when I was a sophomore, and that really awakened me more to biology. And I got eventually more interested in biology than just applied agriculture. I had really strong genetics professor, real good, well-funded background in genetics. But when I was, I think, a senior, I met Dr. Hoge, Terry Hoge. He was a young, energetic cell biologist, trained initially in genetics and statistics, but he had done postdoctoral work in cell biology. And so he had been hired on the faculty of Sam Houston State. And he was the first to bring electron microscopy and some modern technology into that university. And he was, in fact, this is a picture of him in the center there, and that's an old school, really old school electron microscope from about the mid 1960s. He's seated with some of the administrative people of the university. But Terry was a great influence. He was enthusiastic, he was intelligent, but he was serious and a very disciplined scientist. And that's where I got my real start in science, is from Terry and his direction to make it understood. It's exciting, it's fun, but you have to be disciplined, and your work comes first. And he was just great at directing that kind of thought. So I can't tell you that every single day of my life I rely on skills that I learned from Dr. Hoge. He's a really influential guy. So it was a great start.
Wonderful. I mean, getting a mentor when you're a senior and till master's, having the same person guiding you.
Oh, yeah.
That's amazing. And so when you finished your master's and you moved for your PhD, you obtained your PhD, moved to Virginia, you obtained your PhD in 1976. How did you shift from the agriculture major and the cell biology minor to actually getting into developmental biology in PhD? better.
Well, when I studied with Terry Hoag at the master's level, actually, what I was studying was spermatogenesis, but I studied it in invertebrates. I was actually looking at an insect, a grasshopper spermatogenesis. So I began to get interested in developmental biology working with Terry. And at the time, the University of Virginia had a very, very strong department in developmental biology. And one of the people that was in that department was Professor James Norman Dent. And when I attended UVA, I attended there with the idea of trying to work with Dr. Dent, and it worked out. He accepted me in his laboratory. Very interesting. He was a very, very scholarly individual. He was a Fulbright fellow. He had been around. He got his training at Johns Hopkins University, classical embryologist, endocrinologist. You know, in that era, in the 1970s, it was the vogue then was to not use the term embryology. It was to use the term developmental biology. You were old school if you called yourself an embryologist. He had no problem calling himself an embryologist. And he was a classic bow tie wearing, tweed sports coat wearing embryologist, classic. You know, 3 colors of chalk to denote the germ layers. But it was a great education. And not just from Jim Dent's laboratory, but there were just amazing people in that department. I had on my PhD committee of 5 people, 2 of them were National Academy of Science members. So I got to have, my laboratory was next door to Oscar Miller, who was the first to envision gene action with the electron microscope. I had the privilege of visiting with the former chair, Dr. Dietrich Bodenstein. Dr. Bodenstein was a German embryologist. In fact, as a young man, he was in the laboratory of Hans Spemann at the time that they discovered embryonic induction. So I had these really classical people that I was surrounded by, and it was just a fantastic opportunity. Very rigorous program, and I will tell you that that's where I learned science is a 7-day-a-week and including nighttime operation. So we worked full-time. I learned a real good work ethic from these people, but also got a great exposure to a wide variety of contemporary problems in embryology, developmental biology. So it was a good deal.
And what was your thesis topic at that time?
Well, I was studying amphibian. We did amphibian embryology, which of course, a lot of fundamental embryology has come not from necessarily mammals, but from invertebrates, sea urchins, and into amphibians, Xenopus laevis and Rhena pipiens, etc. So we were studying a variety of different things. One of the things that I had studied, and I actually had started with Terry Hoag, there's this interesting phenomenon in amphibians and salamanders. Female salamanders can store sperm. They have an interesting gland where they can pick up sperm from the male, and instead of fertilizing the egg, it goes into this gland. It can be stored for 6 months, 8 months. So the separation of ovulation and picking up sperm is very real. It's also a phenomenon you see in some other animals. So it was very interesting, and we began to study how is sperm stored. We'd love to learn how to do that. But in the process, we looked at the development of other integumentary structures in amphibians, and that's what I ended up doing for my PhD thesis. There's an interesting gland in some amphibians and salamanders that produces pheromones that are really important in reproduction. And very little at the time was known about it. So I ended up being able to isolate and characterizing the biochemistry and of those secretions that are used in trying to adapt to mating. So it was mainly just development of integumentary systems.
Fantastic. I think that was probably like the primary version of chemotaxis that we know now.
Exactly. Yeah.
Works even in humans.
Yeah.
So that's fantastic to know that you began from amphibians and reached human IVF. So after your PhD, after that work, it was homecoming back to Texas, back home. So how was it? How did you stay?
I never expected that. I had no intention of coming back, but it turns out that here in San Antonio there is a pretty well-known cell biologist by the name of Ivan Cameron, and Dr. Cameron had done quite a bit of work on studying the cell cycle, and there's Dr. Cameron. Yeah. And so I really wanted to experience more on a cellular level, fundamental cell processes. So he accepted me as a postdoctoral fellow, and I moved to San Antonio. And in his laboratory, he is a very wide open thinker. And I mean, I really learned, don't close your mind down. Don't, you know, be able to change your mind about things, as follow the data, be able to don't get tied to a hypothesis. And he was really instrumental in promoting that kind of thinking, that be very open-minded, take in everything you can and try to solve problems with a whole host of ideas instead of just being tied to one thought process. And in his laboratory, we became really interested in studying rapidly dividing cell populations and trying to understand the ionic environment within the cells that differed in those kinds of cells compared to those that were no longer dividing. And he had just acquired the technology to do that. We equipped a scanning electron microscope with X-ray detection equipment, so we were able to use that to be able to analyze the ionic basis of rapidly dividing cells. We studied mainly the movement of sodium, potassium, some other elements, and their high levels of sodium in the nuclei of tumor cells and other rapidly dividing populations. So that's where I really gained a lot of expertise in just fundamental cell biology, and that also took us into looking at the physics of water, because that's you know, very primary in moving ions back and forth. And in that, we learned a lot of things to look at water and the interaction of water with surfaces and how it affects ionic composition and whatnot. I think a lot more than I would have if I'd studied with someone else. And that is something I've taken into human embryology and the development of culture technology, macromolecular supplements, and looking at the culture environment. I think I look at it probably a little different way from a lot of people based on those fundamental studies. So.
Because, you know, macromolecular movement is all about the ionic channels. It's all about, you know, the way they move in and out. And I don't think that kind of background can be attained just by doing cell biology. The kind of work you're talking about really looks at, you know, at a micro level, at the sodium potassium channels, for example, yes, that you explained. Yeah.
Right. Yeah. Well, and it also it made me. It made me look at the microenvironment of the human embryo in a much different way. I began to look, if you look in the reproductive tract, it's very interesting. It's not a rich environment for water. It's a moist potential space more so than bulk water. And that really changed my ideas about the way embryos interact with the surface macromolecules in the fallopian tube and whatnot. So I look at it probably a little bit differently than classic reproductive biologists do, but that's why, that kind of a background.
That's fantastic. So I kind of understood like where you come from. You have a background, and not only in cell and developmental biology, but also in looking at a micro level at how molecules flow in and out on the electron transport highway, if I may say so.
Yeah.
So then after this postdoctoral work, you went on to become an assistant professor and get a tenure track from 1978 to 1984. When did you begin research related to human IVF? And who were your collaborators at this time? How did you actually transition?
So when I was an assistant professor beginning in 1978, I got grant funding from the National Institutes of Health, and I was really just studying fundamental cell proliferation and cell differentiation events, looking at somatic cells in culture, and we cultured a whole array of somatic cells to study those processes. But a colleague of mine in the Department of OB-GYN, Ricardo Ash, who was at the university at that time. Ricardo, of course, moved on to California later on and actually got in some trouble. But in those days, he was a young, enthusiastic researcher, and he wanted to start an IVF program. And this was in the very earliest days, in 1980, probably 81 or so, that that came up. And they were having some trouble in the IVF lab that they had tried to set up. So he came to me as a cell culture person to try to work out some of the problems they were having in getting started in IVF. And he was surprised to find out that actually I was trained in classical embryology, which really, he liked that a lot. So I collaborated with Ricardo and his colleague, Jose Balmaceda, and we got an IVF program going. So it was through that. It was actually just an interesting side thing. I wasn't funded to do that. I was being judged on my work in cell biology for promotion and whatnot, but it was so exciting and interesting and new that it was great to get over and toy around in that area, although it was kind of like a hobby almost, that kind of a collaboration. And it was, and the interest in it and the potential in it grew so much that eventually that's why I left and moved full time into human embryology. It's so exciting, and so the potential was just through the roof, much more so than sitting back in a laboratory and growing cultured cells all the time. It was, so that's how it started. Yeah.
Yes, I'm a junior, but I can relate because I moved from PhD back to clinical embryology, so I understand the excitement of seeing a baby.
Oh yeah. Oh yeah.
That's
phenomenal. Yeah.
Yeah. But talking about babies, the first baby that was born was a the cynomolgus monkey through IVF. The world's first and the world's third non-human primate.
Right.
So please tell us how that came about.
Yeah, it was really interesting because the Department of OB-GYN had a non-human primate colony. They had both rhesus monkeys and cynomolgus monkeys, very closely related, but the cynomolgus is a little bit smaller and has a long tail. Rhesus is bigger and has a short tail. In conjunction with developing the human IVF program, Dr. Balmaceda was very interested in doing research with the monkeys and trying to get a research program going. So he invited me to collaborate, and we did that. And so we began to do egg aspirations and learning how to fertilize and culture the embryos from the cynomolgus monkey. And it was tough going. The sperm in these species are very difficult to get them activated, and that's a big part of getting fertilization. So I think a lot of people had tried, but it was very difficult. I had the good fortune of meeting Dr. Barry Bavister. Barry was at the University of Wisconsin at the time, and Barry's lab had produced a live birth with a rhesus monkey. So I talked with Barry's group, and he was very, very kind and altruistic in sharing recipes and ideas and whatnot. And largely through Barry's coaching and whatnot, we were able to get this going very quickly. So we had the first live birth of the cynomolgus monkey. That was the third. Actually, the first live birth was here in San Antonio as well. It was a baboon, and Tom Keel was the guy that produced that. So Tom had one, Barry had one, we had the third one. But at the same time, we were real fortunate here in San Antonio because there is or at least at the time, there's a cattle ranch right outside of San Antonio called Rio Vista Farms. And not only was it a big operating cattle ranch, but they had a big interest in frozen embryos for cattle. So they opened up and built a laboratory, and they were able to recruit Dr. Stanley Lebo to join them. And so Stanley moved to San Antonio, and then we had this opportunity to interact with Stanley and have him as a resource. And that's one of the great things that's happened to me, is to get to know and be real good friends with Stanley Lebo. And then they hired Bill Rawl, and that's the photograph on the left. That's Bill Rawl and Stanley Lebo in their lab at Rio Vista. Bill, of course, was the first one to vitrify embryos, along with Greg Fay. And so we got a lot of coaching and a lot of great ideas from these guys and interacting. It was almost like doing another postdoctoral fellowship to be in Stanley's presence. He was such an enthusiastic man, and he loved to teach and he loved to talk, and we had great collaborative interactions. We've actually ended up publishing a couple of papers together. Stanley passed away about 3 or 4 years ago, but he was at the University of New Orleans, and he did a lot of phenomenal work and interacted with people worldwide. So we were able to apply slow freezing technology to the monkey embryos, and actually we produced a series of frozen embryo transfers, of which we produced the first 2 live births from a non-human primate that resulted from IVF. There had been non-human primates that had been produced from frozen embryos, but those were embryos that had been flushed from the reproductive tract. So we were able to combine these technologies that we've been working on. And to be very honest, A lot of people can tell you. I know Kay Elder is in on this, and Kay could tell you. I look at a lot of things differently from a lot of other embryologists, and I use a lot of technology that's different from a lot of other embryologists. But it stems back to the fact that we had non-human primate embryos that we could work on, and we could do anything to these embryos we wanted to, anything to the eggs that we wanted to, and see what stressed them, what didn't stress them, what worked, didn't work. And really, it's that opportunity of working with non-human primates that has left me in a position to know in the human laboratory, I think, what human eggs can tolerate and what they can't. And there's, I think, some misinformation out there. It's a little bit different than some people think. But anyhow, that was a great, robust model to be able to do those things.
Wonderful. It's so nice to know as to what really went behind the birth, the birth of the first cynomolgus monkey.
Yeah.
And there is just so much science that goes behind doing something, and we just get to see the results. But as you said, you did a lot of fundamental work on what stresses out oocytes, what stresses out embryos.
Right.
And this background in cell culture research and all this experience, how did it really help you design embryo culture media?
Yeah, it fundamentally, we didn't know in the beginning, and I'll be honest, we didn't understand the complexity of metabolism of the human embryo. And we were very lucky because, and David Gardner will tell you that anytime you hear David talk about the early days, he'll tell you the best thing that happened to Louise Brown is she wasn't a sheep, or we probably wouldn't have had Louise Brown. It's true. We just didn't understand the complexity of it, but we also didn't understand that we were getting away with a lot of luck, because the plastic metabolic pathways of the human embryo are not necessarily represented in other species. So we could use a wide array of culture conditions and still get away with it and produce human embryos. There was a marvelous treatise that was produced by Martin Johnson on this subject. And he wrote a wonderful thing about the era of epigenetics and the fact that, yeah, maybe we were lucky, because we still don't know today what we may be doing in culture that may manifest itself 30 years down the road. We just don't know. So it's kind of a curse and kind of a blessing that the human embryo has these very plastic pathways. But it became clear that there were certain conditions that produced a higher quality and more embryos than others. And understanding what you can manipulate in the culture environment, for me, really came from our experience with somatic cell culture. And also the physical way that you can handle eukaryotic cells was, to me, a key in how to handle embryos as well. We had both models to look at to develop those ideas. And it was a very interesting time as we began to unravel that metabolism and the requirements of nutrition for embryos, human embryos, are a little bit different, and they're not somatic cells. And it finally dawned on me that if you think about it from a topological perspective, the human egg and embryo prior to implantation is topologically outside of the body. It is not inside that delimiting membrane that delimits inside from out of the human body.
Yeah.
It's in a unique environment, in a very special place. So I quit thinking about it in terms of what is in the circulation. I mean, a lot of somatic cells you culture based on what's in the circulation or what's in third space. This isn't in neither. This is a unique environment. So that's what really got me to thinking different ideas about what might be required in the microenvironment. And just brilliant work from a whole host of people kind of melded that whole story together. So but having the background is what kind of directed me to thinking about it a little different way.
You basically just talked about epigenetics, and I just wanted to get your view as to, we know that we are getting away from doing certain things in embryo culture and still getting human embryos, still getting these pregnancies. But then there is this use of ICSI that basic scientists like you were saying that, that's tech before science. Science goes in a different way. What's your opinion about this use of ICSI, immediate use of ICSI versus IVF? And shouldn't we do more work on non-human primates and other species to actually look and understand how ICSI should would be changing epigenetics in the species?
Yeah, well, I'm always a big fan of doing things in animal models and being cautious. The problem, of course, is that these problems may not emerge for 30, 40 years, and they may emerge in a way that we're not going to see. That's one of the concerns that I have, for example, in the risk that you take when you're going to transfer mosaic embryos, for example, even what are theoretically low mosaic embryos.
Yeah.
That the baby is born and it has a normal peripheral karyotype in no way means it's not a mosaic, and you may not look at the tissue. Down the road someone has a heart attack and it's because maybe a heart valve is malformed. You're gonna say they died of a heart attack. Well, that may be the location of the mosaicism in thwarting normal formation, and we won't know that. That's the problem. It's a very, very difficult arena. Yeah, but I'm always a huge advocate of looking at what we can in animal models. I will say, I think everybody agrees, human IVF got way ahead of the research in its initiation and its application, and no doubt about that. I would say that cryobiology, though, is a pretty good example of working things out in animals before we got into the scenario with humans. It's not that you can do things carte blanche. I heard a brilliant presentation one time by Steen Willitson, and Steen was very adamant, I think absolutely hit the money with this statement that people kind of feel free to make manipulations to freezing protocols when they have no idea what the consequences of that might be. And I agree with him completely. But I would say from a general perspective that we have millions of calves born from frozen embryos. I mean, millions. To look back on is a pretty good indication that we have a well-founded technology that came from an animal model. IVF wouldn't hold still. People needed it and wanted it. So we're just going to have to live with those consequences, I think, as we learn about them.
I think this is an example where we are going through the long tail of medicine versus the other way around.
Yeah, I agree. Yeah.
Because if in U.S. healthcare, you know that there are two ways, and you definitely understand that this is a very unconventional way of doing healthcare. IVF is a very unconventional field, and you yourself are example of that because you transitioned from a very successful and a very interesting academic career to founding your fertility center. The fertility center of San Antonio. You journeyed from being a researcher to becoming an entrepreneur and an embryologist full time. So we want to know, how did you do this? Because this is something that is still rare, and we want to hear your story.
We went when I was just about at the end of the academic career. I was approached here in San Antonio by a very forward-thinking obstetrician gynecologist by the name of Joe Martin. He had been in academics, and he still had an appointment in obstetrics and gynecology, but he'd moved out into private practice. He was a very successful guy, but he was a very forward-thinking man, and he had a colleague, an ultrasonographer by the name of Kathy Maru, that worked with him, that also wanted to see this technology come outside of the university. The university was losing that technology for a while because Ricardo Ash and Jose Balmaceda moved to California. And while they were rebuilding the university program, there was a little bit of a dearth. So that opened an opportunity, and we made an application to a large general hospital here in San Antonio, and the Methodist Hospital, and they were accepting of the fact, and they gave us an opportunity to establish a hospital-based program, which we did. And that got it going. And again, it was through largely Joe's leadership that allowed us to get that program going. The interesting thing, though, is that Joe was very, very sensitive. He's still a very sensitive guy about what patients are going through, both whatever stresses, whatever medical issues, but also financially. And you do something in a hospital, and the overhead is high. It's expensive. So he was very adamant about getting into a more cost-effective space to increase access to care. And I think he was the first in the state of Texas to move into the private practice arena and establish so we built IVF in an office building across the street from the hospital. And that was a really good move. It let us cut costs. It let us really open up the technology to a lot of the sector of South Texas that wouldn't have probably had it before. So it really was through Joe's leadership. I mean, he's the one that recruited me out of academics, and it was, IVF was so exciting and so enticing. It didn't take much recruiting to do it, but he's actually was the reason that I left academics for that opportunity.
I think patients in Texas and beyond have really benefited from that.
Yeah,
I just wanted, yeah. Yes. So basically, when you actually moved into private practice, this was around the time that many other states were beginning to get into private practice and IVF.
Right.
There's a book by Dr. Howard Jones
that
talks about bringing IVF to America. And he chronicles the moral, ethical problems that you were facing, all of you were facing. How did you personally meet these challenges, and how was the time in Texas and the rest of the U.S.?
Yeah, that's a great question. You know, San Antonio is a wonderful bicultural community. The Hispanic population is actually the dominant population of San Antonio. So Catholicism is huge in San Antonio. In fact, we have downtown still the longest continually practicing Catholic church in the United States, is San Fernando Cathedral. Had a key role in the Battle of the Alamo, which is near and dear to all Texans. But and what happened, as you'll recall, it was 1968 that the Catholic Church published the Humani Vitae, which forbid birth control. Then that subject came back up in the early 1980s. And I will have to say, I think we were really fortunate in the United States to have Howard and Georgiana Jones as the people that kind of were the face of facing that problem. I mean, they are brilliant people, very, very measured, very statesmanlike, but incredibly scholarly people. And they guided us correctly. In San Antonio, you know, in 1987, then February of 1987, the Catholic Church then came down with further instructions, which acquired the name Donum Vitae. And that is where IVF was implicitly forbidden by the church. And the whole rationale for that was, according to the church, it separated the conjugal act from the act of procreation, and that was immoral. Georgiana Jones did a brilliant job, I think, of defending and arguing that position by pointing out that Mother Nature does it herself in the postpartum period and in the menopause. They're separated naturally, so it isn't mankind doing it. The church never really bought into that. So we had problems here in San Antonio when those instructions first came out. There was a bishop in San Antonio that was on television and whatnot that was, you know, pretty adamantly opposed to in vitro fertilization. There was a lot of concern in the general hospital where we were at that time about what effect that would have. I was invited to go on radio programs to debate the Catholic Church, and that was an uncomfortable position because they're good people. They mean well, and you don't want to go on and disparage them. So you kind of had to tiptoe around that a little bit. But by and large, that settled down, and people began to realize that Catholics pretty much make up their own mind about the morality of what they're going to do. So it ended, that kind of settled down, and we didn't have those issues too much anymore. I will say one other event, I was invited to participate in a local Catholic university in a debate, and that whole issue came back up. This is about 2008, 2007, something like that. And I came up against a Monsignor that thought I was the devil. I mean, it was so those feelings still kind of run deep in some people, but in the general community, it's not an issue. We do very well. One of the things that we do, and I'll get off this subject, but we try to tell patients that we are not here to change your mind about anything. I want to know what your beliefs are, so that I can guide the laboratory staff through the process where we're honoring what you want to do. And I think that's really helped us along to just try to treat it as a personal decision of patients.
That was an extremely challenging time that you and all the colleagues and all the seniors went through. Because in India, it's, you know, the population and the government and all the different factions were very accepting when this came. And not as accepting as they are now, but Yes, it should be. It was extremely challenging, I guess, in US when it started, and now they are. It's basically one of the core areas on which we wait for the next research update from US, and we wait for all your papers to come out so that we can go for the next new thing. So when you began your private practice and you were making your own culture media, your own recipes. How did you make your own batches of culture media, and how did you transition from making that to commercial media, and when? And we really want to know about the P1 culture media that you designed and
well,
synthetic serum substitute that you got involved.
Yeah, sure. We made everything. I mean, everything. There was nothing. And that's one of the things where working with the monkey embryos had served me very well. Barry Bavister had actually, going back to his days in graduate school days in Cambridge, had been utilizing Tyrodes, which is a salt solution produced in the early 1900s here in the United States by Maurice Tyrode. He'd used that solution as the base for the media that he was using to culture hamster embryos. And he still used it, a modified form of it, an organically buffered form of it in the monkey work he was doing in Wisconsin. So we began to use that same medium and used it very heavily in our work with the non-human primates. When I set up a human IVF lab, I set up a lab that mimicked what we were doing in non-human primates. I didn't go to another clinical, there weren't too many other clinical IVF labs to go to. So I just did what worked in the monkey, and it worked equally well. So we were making liters and liters of modified Tyrode solution to use for flushing, sperm preps, transfers, etc. I tried a whole array of different culture media. One of the things that got the university program going was the altruistic suggestions and advice of Alan Trouncin. You had him as your first interviewee in the Gurus, and rightfully so, because his altruism really helped the program here in San Antonio. So we got some good advice from what they were using. But in addition, I used some of David Whittingham's media. I used T6, and they all worked to a certain degree. What really changed things was the mid-'80s, when both Eve Azo and Patrick Quinn developed media specifically for human embryos. So when HTF came out and became commercially available in the mid-'80s, we tried it. It worked just as well as anything I made. So that's when we quit making our own medium. The interesting thing about the P1 medium was a medium that was a modification of HTF. But we made modifications based somewhat on what we were doing here and somewhat on what Barry Bavister was doing in his work. And so it was the subtraction of some components, inorganic phosphate and glucose, the addition of amino acids and some energetic substrates that we had been working on here. So it was really just a modification of Patrick's ideas with a lot of Barry's ideas, some of our ideas. We put it together and trialed it, and it worked really well. Because it was really their ideas and their work, I never published a paper on it. I didn't think it was right to take their intellectual property and put my name on it. But Irvine Scientific got interested in commercializing it. And so, and by the way, the P stands for pre-implantation. I was thinking about sequential media. So it was for the first part of pre-implantation development. And I was eventually working on a P2 medium. It doesn't stand for pool. But they got hold of it, and it had some traction, and it was a pretty good medium that worked for a while. It, like the other initial monoculture media, was fine for cleavage, but it absolutely was not good for extended culture. So it had to come in and use it with a sequential pair of second medium or another kind of medium. But that's what precipitated that.
Fantastic, because it was going in parallel with a lot of work that is going in culture media, and we see all these different recipes out, and all of them work and equally well. So,
right. We're in good shape now.
Yeah. So from the time of making your culture media to actually accrediting and auditing laboratories in reproductive programs with CAP, the College of American Pathologists, how did it start in collab with SRBT and ASRM, and what is the state now?
It's really interesting that if you look at federal, in the United States, federal laboratory regulation, the federal regulation that governs all clinical labs is called CLIA, Clinical Laboratory Improvement Amendment. And in the CLIA documents of interest, embryology is not covered by CLIA. And it's still to this day not covered by CLIA. There was some reticence by the reproductive endocrinologists. They didn't want to see it fall into the purview of the federal government. So it did not. And to this day, it did not. So I think that there was some feeling that it would be efficacious if we had some sort of a survey and accreditation program for embryology laboratories. And there was a conversations then that occurred between the American Society for Reproductive Medicine and the College of American Pathologists. And collectively, through a joint interaction they had, they developed the Reproductive Laboratory Accreditation Program, RLAP as it's called. And its initial commissioner was Bill Bird, and it grew from there. And it's still now one of the major accreditation programs. There's another one, it's called the Joint Commission. But it is, the advantage of the program they developed is it's a peer review system. So it's people that are embryologists that are inspecting other embryology labs. And the whole idea behind that is not only to ensure that you're operating under the standards of the college, but it's a collaborative interaction. You exchange ideas. And that's the wonderful thing about the inspection program. I mean, I learn as much from our inspectors as they learn from me, but we exchange ideas and recipes and thoughts and ways to do things. And it's been pretty good. You get some inspectors that are a little more rigid than others, but it's all in all, you know, it can be a very good learning experience. So that's how it evolved, and it's still very strong. Now, the Society of Assisted Reproductive Technology, SART, to be a member of that organization, it's required that your laboratory be certified by either Joint Commission or by CAP. So that's how the program is stabilized, and it's I'd say most of the laboratories in the U.S. are accredited by one of those agencies.
Yes. So, Dr. Poole, we have one question. Is it okay
Sure.
if you take it right now?
Sure.
Okay. So, Dr. Sanjay Shukla from India is asking, for how long, in your opinion, can the oocyte cumulus complexes be kept in HEPA's buffer safely?
Yeah, what a great question. And as long as it's in stable pH, it can stay for an extended period of time, several hours, no problem. And the reason I say that, as long as it's encased in the hyaluronate gel in the cumulus mass, it is largely not seeing its environment. It is insulated. And it is the same principle, exact same principle that you see in amphibians. If you've ever worked with frog eggs, you know they're in a big mass of jelly. And I think there's a biological reason for that, because mom frog doesn't know where she's going to ovulate. It could be in a mud puddle, a horse trough, a pond, a river, a stream, in a laboratory. It really doesn't matter, because those eggs don't see that microenvironment because the encasement of that jelly-like matrix. It blocks, it serves that blocking function. And my idea is that the same thing is true in the human female. She just carries her pond with her in the cul-de-sac, the rectouterine pouch. So I think it serves that same purpose. Well, once you get it out of that environment, out of the follicular environment, and get it into that a well-buffered environment, I think as long as it's a stable pH, it can last longer than a lot of people think. Once you strip it, that's a totally different story. And the thing that most people need to realize is that when you strip an egg, that's when it's the most subjected to fluctuation in pH. It loses its robust mechanisms for modulating intracellular pH transiently during that stage. And so that's a very different story. But as long as it's encased, it's pretty well protected.
I think that's a fantastic input from the side of actually going from animal biology and looking at how it would affect human IVF, the way you were saying that's how you based your whole work on.
Yeah.
So yeah, definitely. All right. So so moving ahead, I'm sorry, I had a fall of the device. So that little threw me off a little. So yeah, moving ahead, you were basically invited by Dr. Bob Edwards, a Nobel laureate and an idol for all of us, to come on the board of the Reproductive Biomedicine Online Journal. So what are your memories of him?
Yeah,
And how
what a phenomenal man. I am so glad Kay Elder is participating today, because she knows Bob Edwards better than just about anybody. In fact, whenever I give a talk that involves that, Kay, I always show the picture of you and Bob at Bourne Hall. It's a wonderful photograph of you two together. I met him. I'm at a small meeting in the very early 90s in California. We were invited to both give talks at this meeting. I was intimidated as could be, because it was just he and I doing the speaking. But what a warm, genuinely great human being he turned out to be. Very, very friendly. We spent, I think, 12 straight hours that afternoon and into the evening talking embryology. And he was he was the most phenomenal resource of information of anyone I ever met in my life. But it was packaged in a very friendly, warm human being. And that's what characterized Bob Edwards. Down the road, about 4 years later, Dr. Rick Paulson from the University of Southern California and I were invited to put on a meeting in Chicago, and we did that, and Bob attended that meeting as well. And that's where I think I really got to know him a little bit better. And it's funny, he came up to me and he said, what would you think if I said that the human embryo by the 2-cell stage has already begun differentiation? Well, I'm sure he already had a long answer to give me when I told him I didn't believe it, but to his surprise, I said, no, I do believe it. And I told him about what happens in the amphibian embryo, because that occurs very, very early in the amphibian embryo. In the frog embryo, the bottom quartet of cells after the third cleavage are the only ones that can make germ cells. The top quartet can't. And there's lots of examples of that. Well, he was amazed to hear that. And he said, listen, I'm working on a paper. Could I send that to you and see what you think about it? I said, flattered beyond belief, of course. Well, like about a week later, this thing arrives in San Antonio. It looked like something had been written by Dostoevsky. I mean, it was as big as the dictionary. And it was a draft of the paper that he wrote with Helen Beard on oocyte polarity, which was a brilliant, brilliant piece of work. So I poured through it and read through it. And I was able to come back and tell him that a lot of the ideas that they were putting forth are really already represented in the animal world in invertebrates, particularly in the small roundworm, Canorhabditis elegans. C. elegans is known genetically, how all of those cells are, every cell in the adult is mapped from the embryo, and a lot of those genetic processes are known. And he wasn't aware of that. He was very open to hearing that, but he wasn't aware of that. So I hooked him up with a C. elegans expert that I knew at Cornell University, and they had a nice discussion about it. But I think that's one of the things that that Bob liked is, although he was a mammalian embryologist, he liked the idea of looking at things across a bunch of phylogeny and looking at conserved mechanisms of reproduction and see how they carry over across different phyla. And I think that's what we got along about. He liked that aspect, that I could bring that to the table. Again, most of the people he dealt with were mammalian people. So that was our real introduction. And then we met at other meetings, and he was just always a warm, wonderful guy. And he emailed one day and invited me to join the editorial board, which was a thrill. I think I spent 15 years on the board there. It was great. Wonderful man. Absolutely wonderful.
What a fantastic, it's just fantastic listening to you sharing how you interacted, because we could actually feel that we are interacting with him. We would never have the option to do that, but you got to interact with this really humble, really scholarly person, and you also, were able to get in his 2 bits of how he thinks that embryo culture is affected.
You had to be on your game with him. Now, he wouldn't let you off the hook. He was a very smart guy. And I will just tell you quickly as an aside, at the meeting in Chicago where Rick Paulson and I put that together, Rick was giving a talk. And Bob stood up and at the question and answer, and he took him to task on something. And Rick was very quick on his feet, and he said, well, that's why we published it in Fertility and Sterility, not Human Reproduction. So he headed it off that way. But no, he's just a brilliant, brilliant man.
Thank you for sharing that. But when we speak about your career, 40 years in IVF and going, and you recently got the SRBT award, Lifetime Achievement Award. How was the experience? And congratulations, you got it SRM 2018. How has it's been so far?
It was the most humbling experience of my life. I don't know why I got that. I honest to this day don't get it, but I'm very humbled by it and very appreciative. Humbled only more by this interview today. I mean, it's very humbling. There are so many great people in our field. I mean, so many very, very good people, and to be put in that category, I have no words for it. I don't know what to say, but just the fact that the rewarding thing is if your peers think that you've done something that's helped them, that takes my breath away. That was really rewarding, very humbling.
It's an extreme honor and an utmost pleasure to hear you speak about your career, and coming back to how family has really moved everything, and how your collaborators have really shaped your career, and how you feel when you speak about them. We can see the passion behind all the work.
Well, that's one of the most unbelievable things, is to have great people to talk to work with, to shape ideas, to bounce ideas off of. And depicted here are three of those. I became very good friends with David Gardner. I think we've been friends 25 or so years. And we've bounced ideas back and forth, and he's been a real inspiration. And we've kind of shared ideas over the past. And so I wanted to depict him. So David on the left, David's on top. His friend there, that was an interesting story. ASRM was here in San Antonio several years ago, and before that, David said, "Hey, could you take me fishing?" I went, "Sure." So that's how this came up. We went down to the Texas coast. In the middle is Dr. Gary Smith. Gary and I have been friends at least that long, and we have worked together on a number of different things, but he's been a great guy to bounce ideas off of and to guide me in thinking. Gary has had some great ideas in using bioengineering in culture platforms and microfluidics. So we shared a lot of ideas about microfluidic technology, which I'm exceedingly high on. I think it's got a real place in embryology. Gary had a great graduate student who finished up, but he wanted him to get some clinical embryology training, and that's Jason Swain. So Jason, after he finished with his PhD with Gary, came to San Antonio and spent 2 years with me, and those were 2 great years. And that's where, in addition to learning clinical embryology, Jason got very interested in pH maintenance and buffer capacity and whatnot. So we did a lot of work together. He did the bulk of the work, let me tell you, but it was just, fantastic work. And so these are 3 of my dearest colleagues, where I still collaborate and interact with all 3, but it just goes to show that you get great ideas from hanging with great people, and these guys are all smarter than I am, so I'll take their advice any day. And I also just have to say I've been blessed to have the family that I have. When you get into this business, the first thing we did when we built the program is I didn't take any days off. For months on end, I didn't have a day off, and you've got a family, a wife that understands that. And my kids have been the same way. I have 2 great kids. I have a son who was actually an animal trainer for about 10 years, a marine animal trainer. He trained whales and sea lions. He's in the IT business now. But I have a daughter that's in the IVF field. She actually was a salesperson for culture media for a couple of years with Life Global. She now works in genetics. She works for iGenomics, and so she's involved. The great thing is, prior to the virus, I could see her at meetings, and so it's been a good experience. But the family's been wonderful. You've got to have that kind of understanding to they have to understand
you work Saturdays, you work Sundays, you work night times. This has to come first. And so it's been fantastic to have that kind of support at home.
Your daughter is in the chat section.
Oh,
She's saying
hi,
hi.
Katie.
Yeah, Katie, it's so nice that you have joined in, because your father was just speaking about you. And it's so lovely to know that you're so connected to your family. And family is the backbone of everything.
Yeah, it is.
And that you do. And I'm sure they are so proud of the career that you have had. Absolutely. And we are so honored to have had you here. Any parting thoughts for us young and aspiring embryologists and anyone who wants to be in the embryology field?
Well, I would tell you this, and you can ask the ladies that work in my lab. I beat on them every day on this, and that is, you can't get around the fact that we're all technologists. You have to be, but you can't let technology drive your life. And that's to me, sort of the sad thing now, is that there's so many things in the IVF laboratory that a young person has to learn how to do. There's so many different protocols now. That there's not much time to look back on our history. There's not much time for the technologists to look and see what's new and coming down the road. And that's, I think, going to be the downfall. I think they're all young scientists, and don't give up on being a scientist. The technology is going to come and go. It's going to change. It just isn't going to much matter. But the science isn't. The science will expand on it. But the fundamentals that guide everything we're doing are the most important. And I think down the road, in addition to wonderful technologies such as artificial intelligence that's coming along, non-invasive technology, but I think down the road, like the embryology lab is still going to play a huge pivotal role in the treatment of patients. I mean, if the idea in modern medicine is to do precision medicine, where we treat patients based upon not just their presentation of their symptoms, but their lifestyle and their individual genetics, so it's personalized medicine, the embryo is the same way. And I can see down the road the lab absolutely doing personalized embryology. Therapeutic culture. I think it's coming. I had really good long conversations about that topic with Michelle Lane prior to her untimely passing, but it's coming. Precision embryology is going to be a wave of the future. So make the science the premier thing, and don't be predominantly a technologist, no matter how hard the pressure is to do that. That would be my advice to anybody. Stay a scientist.
So do you think having university programs that are really focusing on the basics and keeping technology part of it, but not the whole part of it, are they key? What do you feel about education for embryologists?
That's another big problem. In the United States, for example, I just had an email conversation a week or two ago with Rebecca Kreischer about this, and her point that she made is that the fact that the government in the United States will not fund research in human embryology, that's a big part of stifling educational opportunities. We don't have the funding from the government to do that. I would like to see industry take a bigger role in it, because they benefit the most, or they benefit from it. I'd like to see, there's no reason why we can't have that. The way that we're going about things now, I think, is wrong. And that is, if you want an embryologist to come to work for you, the number one way you do that is rob some other program of their trained embryologists. I mean, that's basically what we're doing. We're just offering a better deal and trying to attract somebody away from another program. I'd like to see it where there's educational opportunities, but the fundamentals have to be there in the beginning. Yes, to answer your question directly. I mean, you need to know biochemistry. You need to know some physics. You need to know physiology. You need to know it from a fundamental level. But I think that there could be other ways that we can then train specifically in our field. And I would suggest that it would behoove industry to be involved in that process. And to some extent, we're beginning to see a little bit of that. There are some training opportunities, for example, in different venues around the world where it is industry supplemented. And that's a great thing to see.
Yes, I hope that you get the opportunity to tutor people like us in programs. I would really sit on your classes like the whole day if you were ready to talk, so that we learn really what goes behind animal physiology, right from how macromolecules move in and out, amphibian biology. There's so much that we don't know, and so little that we actually learn on the job. We have to go back and read the theory that you and all the pioneers have got out. That's how we learned, and I hope the next generation gets tutelage from mentors like you.
Thank you very much.
It was an absolute honor, Dr. Thomas Poole, and it was such a pleasure to listen to you talk about your life, the journey, and all the efforts and challenges that went behind making who you are today. And I believe many people who have joined on this webinar have really got inspiration and will further keep getting inspiration from you.
Well, thank you very much. This has been a real honor and a privilege and a pleasure to be with you today.
Thank you.
Thank you. Thank you, Dr. Poole. It was actually wonderful listening to you. We also have a message from Dr. Sanjay Shukla saying, great message from a great man. It's fascinating to learn about your journey in this magnificent field. We appreciate your contribution, sir.
Thank you very much.
We all have really enjoyed the interview. I mean, we don't know how the time went actually. It was really wonderful.
It's been a pleasure. Thank you very much.
Yeah.
Dr. Alex.
Thank you, Dr. Poole. Yeah, thank you, Dr. Poole, being the guru on today's meet. It was really an honor for all of us. We learned a lot of things from your insight, that from a career spanning more than 40 years, and many more years in the basic research. Also, I like very much your mention of your the postdoc mentor and how he shaped the thought process, not to tie down on a particular hypothesis,
right,
but keep eyes open. A great take-home message. And thank you very much, Dr. Poole, once again. And thank you very much, Dr. Stiley. Yeah.
Yes, thank you very much. Thank you so much. And thank you, Alex, for this invitation. Thank you.
Thank you. Thank you.
Thank you, everyone.
Nice
thanks for joining us. And we'll see you next month again with our next guru. Thank you.
Great. Thank you.
Bye. Bye.
It's been an honor. Bye.
Bye.